What if the most sophisticated quantum computer exists inside your cells? Scientists have discovered that DNA may operate using quantum mechanics, the strange physics where particles exist in multiple states simultaneously, essentially making your cells function like miniature supercomputers hiding in plain sight. In 1944, physicist Erwin Schrödinger asked in his book, "What is life?" How could the laws of physics explain living organisms? Today, quantum biology suggests an answer that challenges our fundamental understanding of evolution. Could the mathematical complexity of our genetic code point to something beyond mere chance? The answer might redefine our very understanding of life itself. Schrödinger's revolutionary question. In 1944, while World War II raged across Europe, a renowned physicist was asking a question that would eventually transform our understanding of life itself. Erwin Schrödinger, who had already revolutionized physics with his wave equation, turned his attention to biology. His lectures at Trinity College Dublin, later published as the book What is Life?, posed a question that seemed simple but carried profound implications. How could the physical laws that govern atoms explain the behavior of living organisms? Schrödinger recognized a fundamental problem. Classical physics, the physics of Newton that explained the motion of planets and objects we can see, failed to explain how genetic information could remain stable across generations. At a time when genes were still a somewhat abstract concept and the structure of DNA remained unknown, Schrödinger made a remarkable intuitive leap. Approaching biology through the lens of quantum mechanics, the strange new physics he had helped create, Schrödinger proposed that genetic material must be structured as an aperiodic crystal, a stable molecular structure containing vast information in its irregular arrangement. This prediction came 9 years before Watson and Crick discovered DNA's double helix structure. Like a bridge spanning two distant shores, Schrödinger connected the mathematical world of physics with the organic complexity of biology, creating a pathway future scientists would follow. In the 1940s, biology and physics existed as separate disciplines with little crossover. Biologists studied whole organisms using microscopes and dissection, while physicists worked with equations and particle accelerators. The application of quantum physics to living cells seemed far-fetched to scientists in both fields. Schrödinger was creating a new scientific territory. He wrote that understanding life would require new physics, not because existing physics was wrong, but because biology presented challenges that would expand our understanding of physical laws. This interdisciplinary approach predated by decades the merging of fields we now take for granted. The reception to his ideas mixed skepticism with fascination. Many biologists dismissed his theories as speculative overreach from a physicist unfamiliar with living systems. Some physicists worried that applying quantum mechanics to biology would undermine the mathematical rigor of their field. Yet others were intrigued by the questions he raised. What made Schrödinger's work so prescient was his specific predictions about the molecular basis of heredity. He correctly reasoned that genetic information must be encoded in a molecule stable enough to preserve information over generations, yet variable enough to allow for the diversity of life. He estimated how much information such a molecule would need to contain and speculated about its structural properties. When Watson and Crick discovered DNA's structure in 1953, the molecule matched Schrödinger's conceptual description with remarkable accuracy. DNA is indeed an aperiodic crystal with a variable sequence of bases encoding genetic information. The double helix contains billions of chemical letters forming the genetic instructions for building and operating living organisms. Schrödinger had been right about information storage too. He recognized that life requires a physical system capable of containing enormous amounts of information in a stable form. DNA's four-letter alphabet provides this capability, with a single human cell containing about 700 megabytes of genetic information. Despite these insights, technological limitations prevented scientists from testing whether quantum effects actually operate in biological structures. The instruments needed to detect quantum phenomena in complex molecules simply didn't exist. Experimental verification of his hypothesis would wait decades. Through the following decades, molecular biology advanced rapidly with discoveries about DNA transcription, RNA, and protein synthesis. This central dogma seemed to explain life's fundamental processes without needing to invoke quantum mechanics beyond its basic role in chemical bonds. Schrödinger's more radical suggestions about quantum effects in biology faded into the background. The technological barriers were significant. Quantum effects occur at extremely small scales and last for incredibly brief periods, often less than a billionth of a second. Detection requires specialized equipment that wasn't developed until the late 20th and early 21st centuries. Schrödinger made these predictions without modern computational tools. Today, scientists use supercomputers to model quantum interactions in biological systems. He worked with pen, paper, and mathematical intuition developed through his fundamental physics work. His insights extended beyond genetic material structure. Schrödinger questioned how enzymes could catalyze chemical reactions with such precision and efficiency, suggesting quantum mechanics might explain these biological catalysts, another prediction modern research is now confirming. It took nearly 60 years for technology to advance enough to begin testing Schrödinger's hypotheses about quantum biology. With ultra-fast lasers, sensitive spectroscopy, and quantum sensors, researchers began detecting phenomena like quantum coherence, tunneling, and entanglement in living organisms. Evidence now confirms Schrödinger's suspicion: DNA functions as a quantum system in specific ways. The electron arrangement in DNA bases creates conditions where quantum effects influence mutations, DNA repair, and possibly information storage. The hydrogen bonds holding DNA's double helix together exhibit quantum properties affecting the genetic code stability. Schrödinger's question, what is life? Opened a door scientists continue to explore. His suggestion that quantum mechanics plays a fundamental role in biology launched what became quantum biology, examining how quantum effects influence processes from photosynthesis to bird navigation, enzyme function, and possibly consciousness itself. These discoveries are transforming our understanding of biology at its most fundamental level. The quantum nature of life that Schrödinger proposed is moving from theoretical speculation to experimental reality, opening new frontiers in both biology and physics. This convergence raises profound questions about life's nature and origin, revealing that plants and other organisms have been harnessing quantum effects all along. The quantum nature of photosynthesis. Plants across our planet perform a remarkable feat every day that may rely on quantum mechanics. Photosynthesis, the process that converts sunlight into usable energy, operates with an efficiency that has puzzled scientists for decades. The question emerges, have plants been using quantum computing principles to harvest sunlight for billions of years? Photosynthesis stands as perhaps the most fundamental biological process on Earth. Without it, the oxygen-rich atmosphere that supports animal life wouldn't exist. Plants convert incoming sunlight to chemical energy with efficiency rates approaching 95% in the initial energy transfer steps. This level of efficiency exceeds what human-engineered solar panels can achieve, which typically convert only 15 to 20% of sunlight into electricity. For most of scientific history, researchers understood photosynthesis through classical physics models. The dominant theory, known as force-through-resonance energy transfer, FRET, explained how energy moves from molecule to molecule when a chlorophyll molecule absorbs a photon of light. While this model explained basic observations, it couldn't account for the extraordinary efficiency rates. Classical physics predicted far more energy loss as heat during these transfers than what scientists observed. This efficiency puzzle led researchers to suspect something beyond classical physics must be happening. Plants somehow avoid the random, wasteful pathways that energy would typically take. It was as if the energy identified the most efficient path rather than bouncing around randomly. In 2007, a groundbreaking nature study provided evidence challenging the classical understanding. Scientists studying the phenomenon of Diffusion FMO, complex in green sulfur bacteria, discovered something extraordinary. This protein complex serves as a molecular wire conducting energy from light-gathering antennas to the reaction center where chemical conversion occurs. The FMO complex consists of 3 identical protein subunits, each containing 8 precisely arranged bacteria chlorophyll molecules. These molecules form a molecular circuit board with their specific positioning critical for the quantum effects that researchers discovered. The study revealed that excited energy states in the FMO complex didn't simply hop sequentially from molecule to molecule. Instead, the energy existed in multiple locations simultaneously, a phenomenon called quantum coherence. This mirrors quantum behavior that enables quantum computers, where quantum bits exist in multiple states simultaneously rather than just binary 0 or 1. Using femtosecond multidimensional spectroscopy, employing incredibly short laser pulses, researchers tracked nearly instantaneous energy movements. These experiments revealed quantum beating, oscillations indicating quantum coherence. Remarkably, this coherence persisted for up to 660 femtoseconds at room temperature, far longer than theories predicted possible in biological environments. The energy in photosynthesis behaves like a driver who can test all possible routes on a highway simultaneously before instantly materializing on the fastest path. Rather than trying each path sequentially like classical navigation, the energy wave explores all paths in parallel before selecting the optimal route, essentially performing a quantum computation to maximize efficiency. What makes this discovery astonishing is that quantum coherence was thought to require highly controlled environments, near absolute zero temperatures in a vacuum. Biological systems with their warm, wet, constantly moving molecules seemed incompatible with quantum effects. Yet, evidence shows the protein structure might actually protect and enhance these quantum effects rather than destroying them. The protein scaffold vibrations appear tuned to the right frequencies to support quantum coherence. Imagine an orchestra where molecular vibrations play in perfect harmony, creating a protective environment for quantum processes rather than disrupting them. Evolution seems to have optimized these proteins for both their chemical and quantum mechanical properties. Similar quantum phenomena have been observed in various photosynthetic complexes across different species, from ancient cyanobacteria to modern plants. This suggests quantum mechanics is a widespread strategy in nature for efficient energy capture and transfer. These discoveries have sparked new research directions in both quantum physics and biology. Engineers now study these natural quantum systems to design better solar cells and energy technologies. If we can replicate nature's quantum methods, we might develop artificial photosynthesis systems with similarly high efficiency. The discovery of quantum effects in photosynthesis challenges our understanding of evolution. Has natural selection been optimizing quantum processes for billions of years, long before humans understood quantum mechanics existed? This finding breaks down the conceptual barrier between quantum and biological worlds. The warm, wet environment of living cells no longer seems incompatible with quantum effects. This insight has opened investigations into other potential quantum phenomena in biology, from enzyme catalysis to DNA mutation mechanisms. The quantum nature of photosynthesis suggests life may be intrinsically quantum mechanical at its core. Plants haven't been using an analog of quantum computing, they've been performing actual quantum computations in their cells for billions of years. As we continue exploring quantum foundations of biology, another surprising area has emerged. The role quantum mechanics plays in how enzymes work, particularly in DNA itself. Quantum tunneling in enzymes. Inside your body right now, billions of chemical reactions are occurring that, according to classical physics, should be impossible. These reactions happen not because the laws of physics are being broken, but because they're being bypassed through a quantum mechanical phenomenon called tunneling. This process is particularly crucial in enzymes, the biological catalysts that make life possible. Enzymes are specialized proteins that accelerate chemical reactions in living organisms. Without these molecular machines, the reactions necessary for life would proceed too slowly to sustain biological functions. Your body contains thousands of different enzymes, each designed to facilitate specific chemical transformations. They're responsible for everything from breaking down the food you eat to replicating your DNA when cells divide. Enzymes increase reaction rates by factors of millions or even billions compared to uncatalyzed reactions. This acceleration allows processes that would naturally take years to occur in fractions of a second. Understanding how enzymes achieve this remarkable efficiency has puzzled scientists for decades. Chemical reactions require molecules to overcome energy barrier, like climbing a mountain before transforming into products. This barrier, the activation energy, determines reaction speed. Traditional enzyme theory explains that enzymes lower this barrier by holding reactants in optimal positions. However, this explanation only partially accounts for enzyme efficiency. Scientists consistently observed reactions occurring hundreds of times faster than classical models predicted, particularly for hydrogen atom transfers common in biological systems. The solution came quantum mechanics and the phenomenon of tunneling. In the quantum world, particles exist as probability waves rather than definite objects. This wave-like nature allows particles to occasionally pass through energy barriers instead of going over them. Imagine a shortcut through the mountain rather than climbing over it. Quantum tunneling creates these impossible pathways. Hydrogen atoms, being the lightest elements, exhibit the strongest tunneling effects. Their small mass gives them a more pronounced wave-like character, increasing their tunneling ability. This property becomes critical in many enzyme reactions involving hydrogen transfers. In 1989, scientists at MIT conducted a groundbreaking experiment with the enzyme alcohol dehydrogenase, which breaks down alcohol in your liver. They replaced normal hydrogen atoms with deuterium, a heavier hydrogen isotope. If classical physics alone governed the reaction, this substitution would cause only minor slowdowns. Instead, they observed reaction rates dropping dramatically by factors of 50 or more. This provided compelling evidence that quantum tunneling was essential to the enzyme's function, as the heavier deuterium atoms couldn't tunnel as effectively as hydrogen. DNA polymerase demonstrates this quantum advantage remarkably. This enzyme replicates DNA with an error rate of less than 1 in a billion. Like a quantum courier taking shortcuts through an impossibly congested city, hydrogen atoms tunnel between DNA bases, allowing the enzyme to check and correct potential errors with extraordinary speed and precision. Without tunneling, DNA replication would be too slow and error-prone to maintain genetic stability. Scientists have used advanced spectroscopic techniques to directly observe hydrogen tunneling in enzymatic reactions. These methods track atom positions with extraordinary precision, showing that hydrogen atoms don't follow classical paths, but instead tunnel through energy barriers. Enzymes haven't just passively adapted to quantum effects, they've evolved to optimize them. Their structures create environments that position donor and acceptor atoms at ideal distances for tunneling. Protein dynamics, subtle movements within enzyme structures, bring reactants into alignments that maximize tunneling probability. This fine-tuning suggests that quantum effects aren't incidental, but essential to enzyme function. Beyond DNA polymerase, quantum tunneling appears in enzymes involved in cellular respiration, photosynthesis, and various metabolic pathways. Soybean lipoxygenase shows some of the most dramatic tunneling effects ever measured in a biological system, accelerating hydrogen transfer rates by over 1,000-fold compared to classical predictions. Quantum tunneling contributes to both DNA mutation and repair. Occasionally, hydrogen atoms and DNA spontaneously shift positions through quantum effects, creating tautomeric forms, alternative arrangements of the same atoms that can lead to mismatches during replication. These quantum events contribute to the background mutation rate driving evolution. Repair enzymes then use tunneling themselves to identify and fix these changes, creating a balance between genetic stability and evolutionary variation. This quantum view transforms our understanding of life at a fundamental level. Biological systems have evolved to harness and control quantum effects. The precision of DNA replication, the efficiency of metabolic processes, and ultimately the stability of the genetic code all depend on quantum tunneling. The discovery that life depends on quantum effects raises profound questions about how such systems evolved. The enzyme structures facilitating quantum tunneling require precise atomic arrangements encoded in DNA, which itself relies on quantum tunneling. This creates a remarkable interdependence where quantum effects enable the very systems that have evolved to harness them. When considering life's origin, we must account for the emergence of structures optimized for quantum mechanical processes. This adds significant complexity to mathematical models calculating the probability of life arising through random processes. As we'll see next, when these quantum processes are incorporated into calculations about DNA's formation, the mathematical picture becomes considerably more intricate. The mathematical improbability debate. The mathematical challenge of calculating the probability of life's emergence has fascinated scientists and philosophers for generations. Now, with our growing understanding of quantum biology, this calculation has become significantly more complex. When we try to determine the odds of quantum-capable DNA forming by chance, we enter mathematical territory that stretches the limits of probability theory. Traditional biological structures are already remarkably intricate. A single human cell contains approximately 30,000 different proteins, each folded into precise three-dimensional shapes necessary for their function. DNA itself consists of billions of nucleotide bases arranged in a specific sequence. The probability of such complex structures forming randomly has long been debated in evolutionary biology. Quantum biology adds additional layers of complexity to these calculations. The physical arrangement of atoms and molecules matters critically alongside their quantum mechanical properties. For DNA to function as a quantum system, it requires precise arrangements of electron clouds, specific distances between atoms that enable tunneling, and molecular structures that can maintain quantum coherence. These quantum requirements significantly constrain the possible arrangements that would result in functional biological systems. Consider finding a specific grain of sand on all the beaches of the world. This gives us a glimpse of the probabilities involved in forming complex biological structures randomly. Several mathematical models have been used to calculate these odds. The simplest approach multiplies the individual probabilities of each component forming correctly. For a moderate-sized protein of 300 amino acids, with 20 possible amino acids at each position, this calculation yields a probability of 1 in 20 to the power of 300, an astronomically small number. When quantum requirements enter the equation, the probabilities shrink further. Quantum tunneling in enzymes requires atoms positioned at precise distances and angles. Maintaining quantum coherence demands molecular structures that shield quantum states from environmental decoherence. These constraints dramatically reduce the mathematical probability of such systems forming by chance alone. This mathematical territory has become central to debates about life's origins. Proponents of intelligent design highlight these calculations as evidence for their perspective. They point to probabilities like 1 in 10 to the power of 183 for specific protein sequences forming randomly, concluding that such improbability points to deliberate design rather than chance. However, evolutionary biologists offer substantial counterarguments that transform the probability landscape. Natural selection fundamentally changes these calculations. While mutations occur randomly, selection pressures consistently favor certain outcomes, making evolution a non-random process. Instead of requiring all components to form simultaneously by chance, evolution works through incremental steps, each providing some survival advantage. The vast time frames available for evolutionary processes also alter probability assessments. Life's 3.8 billion year history on Earth provides enormous time for evolutionary exploration. With trillions of organisms existing simultaneously, each potentially experiencing mutations, the probability space has been extensively sampled. What seems improbable in human timeframes becomes more reasonable over geological time. Selection pressure makes improbable structures achievable through natural processes. Even small improvements in function can provide survival advantages. Once a basic function emerges, selection can rapidly refine it. This directed optimization means evolution doesn't blindly search the entire possibility space. It follows paths of increasing fitness, significantly improving the odds of complex structures forming. Computer simulations confirm this, demonstrating that when selection is applied after each random step, complex structures reliably develop from simple starting conditions. These models suggest quantum-capable biological systems could evolve through similar processes of incremental improvement under selection pressure. A critical error in probability calculations involves what researchers call the post hoc probability fallacy. This occurs when calculating the probability of a specific outcome after it has already been observed, like calculating the odds of a specific protein sequence we see today.